Hello, welcome to NPTEL certification online course on Pharmacognosy and Metabolic Engineering. So, this is lecture 9 where I will discuss plant genetic transformation. Let us go to the next slide to understand why it is needed. So, the concepts what will be covered here, that why plant genetic manipulation is necessary, cell biology of agrobacterium mediated natural gene transfer. So, under this we will talk briefly about Ti plasmid and tDNA, mechanism of tDNA nicking, transfer of tDNA from bacterium to plant cell and integration of tDNA into plant genome. The first question is that why genetic manipulation is needed. So, what is clear to you that this course basically we are dealing with the formation of specialized metabolites or natural products which were useful to us either for medicinal purpose or for different other purposes. So, most important part is we are dealing with the product. Now, product what is showing here, the product is formed mostly as a result of enzyme catalyzed reaction. So, enzyme is a protein. So, and the reaction means it requires a substrate. So, the substrate is again another natural product. So, that substrate through enzyme catalysis got converted into the product. Now, the enzyme protein which is basically is a part of the central dogma of molecular biology, what it says that DNA makes RNA, RNA makes protein. So, there is a gene which codes for a particular protein which is say here. So, the gene first will undergo transcription, as a result of that messenger RNA will be produced and that messenger RNA will subsequently translated into the protein and that protein may undergo certain post translational changes to become active and then that protein, enzyme protein, if it gets the right substrate, then it will perform its catalytic reaction and as a result of the product is formed. The purpose of genetic manipulation is because we want to insert the specific gene for a specific protein, so that the desired product can be made. That is why the genetic manipulation is needed, and here the source of the gene source could be the same plant or another plant or maybe from fungus or maybe from bacterium. So, either homologous or heterologous origin. So, that has to be transferred within the plant cell, and what we are going to see, what is the most reliable method of transferring this gene into the target plants. There are different methods of gene transfer available, but we cannot really discuss all this because that is not the focus of the course. So, that is why I mentioned the most reliable method we will discuss. Let us go to the next slide. So, genetic manipulation. So, source of gene, let us say this plant from where the source gene will be taken, that is the gene of interest. That gene will be isolated using the molecular biology methods. Okay. And as I said in the previous class that this part is not covered. So, from our part covered actually once we have the gene in hand, then how the gene will be transferred into the target plant. So, this is the target plant and you see this is the gene of interest and that which is denoted here in the middle, and that is now processed using a proper vector and then it is transferred into the plant, and the target plant this gene is integrated within the nuclear genome, and if it is integrated there, then it is expected then that it should function properly. So, the most reliable method of transfer gene into plants is using a natural genetic engineer, which is nothing but a soil bacterium, which is a gram negative soil bacterium, which is called Agrobacterium tumefaciens. This soil bacterium infects mostly dicot, and upon its infection, how it infects, the it infects through the wounding site of the plant, and it transfers a part of its plasmid DNA into through the wounding site into the host plant genome, and upon transfer this part of the plasmid DNA, once it is integrated within the host plant genome, it modifies the hormonal balance of the host plant, and as a result what we see the crown gall formation, which is the external manifestation. So, the crown gall, so that is why this is called crown gall disease. It is itself is a disease, and the crown gall first detected in the cherry plant, and subsequently the tumor inducing properties were worked out. So, the crown gall disease first recorded also from grapes in 1853, and the scientists they found the bacterium which is responsible for the crown gall disease. So, that bacterium is nothing but now called it as Agrobacterium tumefaciens, because it induces the tumor. This is a typical crown gall tissue. And important point to note here that crown gall induces growth at the wound site and severely limits the crop yield as well as growth vigor. That is why crown gall itself is a nuisance. Now, why crown gall happens? So, two scientists Edwin Smith and C.O. Townsend, they started working into the issue, and from the gall they have isolated the bacterium. So, which is Agrobacterium tumefaciens, and this bacterium they isolated, and then they obtained pure culture, and then from this pure culture they inoculated this bacterium into a fresh plant, and what happens now the fresh plant started producing started making this gall. That means, the property of the gall formation lies with the bacterium. So, what is there? If you take a normal plant and put it in a medium without any addition of hormone, the plant will not survive for a long time. However, if you add hormones such as auxin and cytokinin, then the tissue grows and survive, and it will continue to grow as long as you put the proper medium with a balanced hormones. Now, if you take the crown gall tissue and add it into the medium, and here if you do not add hormones, what is going to happen? The things which is what is going to happen is this, that crown gall will continue to grow. That means, for the growth of crown gall there is no need of external supplementation of auxin and cytokinin in the medium. That means, the cell, the crown gall cell has the capacity for autonomous growth, and that is as a result of permanent activation of growth substance synthesizing systems. That is what A.C. Brown mentioned in 1958. So, that means, the properties of synthesizing growth substances are already acquired by the crown gall tissue, but not in the normal callus tissue. Now, if you see, if you as I said that they have isolated the bacterium for and further study what they revealed, that this Agrobacterium tumefaciens contains a large plasmid in addition to its genomic DNA. So, that plasmid is called TI plasmid, that means tumor inducing plasmid. So, now if you look into details of this plasmid, there are two regions. One is what is showing in the red, that is called tDNA or transferred DNA. The term transfer itself indicates that this part of the DNA is transferred from the Agrobacterium through the wounding site to the plant cell, and then it travelled through the cytoplasm and then it ultimately reach the nucleus, it enters into the nucleus and then it become integrated as a part of the with the plant genome. In addition to transfer DNA, there is another region which is called virulence region. So, virulence region is much larger in size compared to the tDNA, and this region contains several genes which are called virulent genes. In fact, all these virulent genes act in operon. So, there are several virulent genes which coordinate and function together, and the main function is basically to make the tDNA or transfer DNA cut from the agrobacterium plasmid, and it will guide the tDNA to go out from the agrobacterium cell. And how it is going out? So, it forms a channel which connects the agrobacterium cell and the plant cell. That means, it forms a pipe. Through the pipe, the tDNA safely moves from agrobacterium and enters into the plant cell. So, that is all done by the virulence gene product, and the pipe what I said, that is made up of protein only. And not only that, so it is also being guided. So, it is guided from the agrobacterium till the transfer DNA enters into the plant nucleus. So, that means virulence plays very important role in transfer of the tDNA from agrobacterium into the plants. If you see further details of the TI plasmid, what you see that the transfer DNA or tDNA, it contains two regions, one is called left border, another is called right border, and these are 23 to 24 base pairs imperfect repeats, and this actually demarcates the region which will transfer from the TI plasmid into the plant from the remaining region. So, it contains the genes for auxin synthesis, genes for cytokinin synthesis, genes for opine synthesis. So, opines are unusual amino acids which are produced by the agrobacterium to support its own growth, and outside that we have this Vir gene cascades, and then there is a origin of replication that allows agrobacterium to replicate, and there is a region which is called the genes for the opine catabolism. So, that will help to utilize the opine, and then there are other regions for conjugal gene transfer. So, this all makes the TI plasmid. Next, let us see. Now, this is a very simplified diagram. As you see that a TI plasmid, it contains the tDNA flanked by two border regions, and now this is cut, and this tDNA now moves inside the plant cell, and this yellow arrow is nothing but the pipe like structure which is formed by virulence B complex, virulence B proteins. So, that form this structure through which the tDNA safely enters into the plant cell. So, as I mentioned that opine synthesis to feed the agrobacterium, and these regions auxin and cytokinin, and once this tDNA get integrated into the plant cell, and then what it happens, because it has the property for auxin and cytokinin synthesis, and that is why if the crown gall tissues can grow in the medium if you grow it under in vitro condition, and because it gets the support of both auxin and cytokinin from the DNA, crown gall DNA, which basically acquired from the agrobacterium itself, and that leads to the autonomous growth. This is what inside the black box, how agrobacterium transfers tDNA. So, first of all, the agrobacterium lives in the soil, and in the plant the roots are embedded there, and what happens that wounding happens in the root. Upon wounding, what happens, the plant basically release certain chemical substances. So, one such substances is called acetosyringine. Actually, that substance basically activates the agrobacterium's virulence cascades, what those agrobacterium which are living near the plant. Okay. So, it activates the virulence. So, first the vir A is activated, virulence A. I have not put all details here, just only I am telling verbally, that activates the virulence G, which subsequent activates the other virulence genes such as vir D1, vir D2, vir D3, then vir E, vir F, and of course, vir B. So, all this plays important role. For example, vir D1, vir D2, vir D3, that plays important role to cut the tDNA from the Agrobacterium TI plasmid, and then it also make another cut the other side. So, first there will be the cut in the right border, then there will be cut in the left border, and then once this portion is excised, then it Agrobacterium through the DNA repairing mechanism, it repairs its plasmid. And the cut DNA, what happens, the tDNA. So, one vir protein which is vir D2, which is attached in the other 5 prime end, which is basically guiding the tDNA to move out from the Agrobacterium through this tunnel and enters into the plant cell. Now, one important point is this, the cytosol of Agrobacterium, which is basically a prokaryotic, and the cytosol of plant cell eukaryotic. So, there are a lot of proteins present, there are a lot of proteases present, and there are maybe other DNA nicking enzymes present. So, in fact, Agrobacterium has to transport inside the plant cell, and which is not always very favorable for its smooth passage. So, the Agrobacterium single stranded tDNA, which is nicked out, will be wrapped by another vir protein, which is called vir E2. So, vir E2 basically will protects the Agrobacterium tDNA from all sort of hazardous elements which is present inside the plant cell, and then this enters, and there may be certain proteins of the plant as well as certain other vir proteins which guide the single stranded tDNA to move inside the nucleus through the nuclear pore, and once it enters. So, it try to integrate in the euchromatin region, and because euchromatin region are loosely arranged, heterochromatin is very tightly packed, and then once it integrates properly, and moreover euchromatic region is the transcriptional active sites. So, once it integrates properly, then it will become a part of the plant DNA. Once it becomes the part of the plant DNA, it now dictate the plant cell to do the job what it wants. So, what is the job? It basically asks the plant cell to alter its hormonal balance, because the tDNA contains the genes for auxin and cytokinin as well as opine. So, plant cell is producing these hormones in higher amounts, and as a result of that you will see changes in the plant growth. So, once the tDNA enters into the plant, so this basically a simple model, Tinland's model, which explains that how this pink color single stranded Agrobacterium tDNA integrated into the plant genome. So, what is showing in the blue is basically the double stranded plant genomic DNA. So, and these triangle things are nothing but these are the cut site. So, you see cut 1, cut 2, there is a cut here as well, and what happens that the first the Agrobacterium tDNA finds a small region on homology where it makes base pairing, and it is here, and then the excess region is basically cut out, and still the vir D2 is attached. So, in the next phase what happens that there will be cut in the plant DNA. So, that is showing 1, and then this Agrobacterium will now join with the plant DNA, and in the meantime agrobacterium try to make base pairing with the plant DNA. So, there will be another cut here and there may be one single nucleotide pairing will be sufficient and then it hangs here. So, at that point of time VD2 is removed and once it cuts the plant DNA. So, this is the displaced plant DNA which is also removed and as you see that this is now if joins then this becomes a part of the plant DNA. So, what is required basically it requires there is one more cut which should be here and here in the other strand. So, this will be empty. So, there will be now as it is shown in this small arrow that this strand will start synthesizing its second and then it basically joins here. So, the single stranded agrobacterium with the help of plant enzyme it now makes the double stranded form and then it is totally integrated into the plant genome. So, now what we see the single stranded agrobacterium tDNA which enters now it is properly integrated and it is becoming double stranded and it becomes a part of the plant DNA. So, let us see this in the form of a summary that this is the tDNA which contains left border and right border and as I said that the plant basically wounding it releases phenolic compounds that activates the virulence A which in turn activates the virulence G through phosphorylation that in turn activates the other virulence genes in the tDNA and as a result of that what we see is this this is the induction of other VIT genes and then what we see in the next step is this once this is induced that means it produces the VIT D1, D2, D3 then it started the processing of tDNA. So, the D2 is important I said that once it cut it joins it basically binds at the 5 prime end and then other VIR protein comes into the picture. So, D2 and then E2, E3, F these all plays important role in transferring the tDNA from to move out from agrobacterium and enter into the cells and T4SS is basically it is basically forming the that pipe like structure through which the agrobacterium with D2 and E2 attached moves out and enter into the plant and once it enters you see that D2 and wrapped by E2 and then E3 and F they are guiding. So, they may have a nuclear localization signal. So, it is guiding so that the tDNA reaches a proper near a proper nuclear pore and through which it enters into the nucleus. There are certain plant proteins which also helps to enter this agrobacterium tDNA inside the plant nucleus and then finally, it is integrated into the plant nucleus and if it is integrated in transcriptional active sites then it will transcribe and the gene expression happens in the tDNA for auxin, cytokinin and opines and as a result we will see the crown gall formation. So, what is the lesson we learned from here? The lesson we learned from here if we go back to the previous slide that this is a natural gene transfer mechanism. That is why agrobacterium is called natural genetic engineer. So, agrobacterium transfer a part of its plasmid DNA into the plant, so that it transfer it utilizes the nutrients from the plant cell for its growth. So, if you see the crown gall tissues it contains lot of agrobacterium and not only that the opines are needed for the agrobacterium growth. Now, the tDNA contains gene for opine synthesis, that is why once it is integrated into the plant it is directing the plant to produce the opine. So, that agrobacterium happily live and propagate there. Now, this is a natural gene transfer mechanism. Now, if we go back to our original intention is that to increase the content of the natural products in the target plants by transferring the desired DNA into the target plant, and that desired DNA means which is the gene, and if the gene is properly expressed, then it will make the functional protein, which is basically nothing but the enzyme, and if it gets substrate, it will produce the product. So, how we can connect that event with this event? As I have mentioned that this is the one of the most reliable method, rather most reliable method of transfer DNA into plants. So, this is a natural genetic engineering mechanism perfected over the evolution. Now, if you have the piece of DNA and if you put it here between the left border and right border, then what is going to happen? That piece of DNA which is a part of the gene. So, when it transfer its tDNA, the gene also get transferred through agrobacterium into the plant cell, and as the mechanism is very precise, you can be very sure that gene transfer happens and it integrated into the nucleus. So, it will not go to the mitochondria, not it will go to the plastid, or not it will go to any other organelle, or it gets degraded. So, such a perfected mechanism must be harnessed to transfer the gene of interest into the plants, and agrobacterium is the perfect vehicle for doing that. So, here lies the beauty, that is why I have discussed the cell biology of agrobacterium plant interaction and agrobacterium mediated tDNA transfer. So, this knowledge in the subsequent class we will utilize for making specific vectors to transfer gene of interest into plants. Now, this species is Agrobacterium tumefaciens because it produces crown gall tumor. There is another species which is also available, which is called Agrobacterium rhizogenes, which produces hairy root disease. So, it has a similar plasmid makeup. So, here we call tDNA, there we will call RI plasmid, here we call TI plasmid containing tDNA. So, there will be RI plasmid containing tDNA. RI means root inducing plasmid, and I have mentioned hairy root in previous classes. So, if you take a natural wild type Agrobacterium rhizogenes and infect your plant tissue, and if Agrobacterium is capable of transferring its natural tDNA which resides inside the RI plasmid, it will make the hairy roots. So, I will talk about that also in the next class, and we will also talk about that how specific genes can also transfer into the hairy root. So, we will more talk about the transformation, co-transformation and vector construction, so that a successful gene transfer can be made to fulfill the desired goal of our natural product biotechnology. So, in conclusion, Agrobacterium is an amazing organism with a unique ability to transfer DNA into diverse host genome, which has been exploited to facilitate research and breeding. And Agrobacterium research and its application went far beyond what Smith and Townsend could foresee when they found crown gall was caused by the bacterium 1907. So, with this I end the class today, and thank you very much.